Okay, this was a tricky exam. The June was nicer. So let's go to the first question. That was an easy question. Okay, we will not read but you will. Here, the Golgi body, where is it found? Hmm? It's found, is it found in the prokaryotic? Definitely not. So, I like to scribble. And the circular DNA? Yes. So, the Golgi body is found in the eukaryotic cells, but not in the prokaryotic cells. Circular DNA is found in the eukaryotic cells, where? In the mitochondria or in the chloroplast, if we are talking about a plant cell. So it is found in the eukaryotic cells, and also it is found in the prokaryotic nur, or what, or not? Of course, yes. Okay, prokaryotic cells are bacterial cells. They have no nucleus, but they have DNA. Okay, they have circular DNA. This is in any bacteria, and sometimes they have extra genes in a plasmid. And this plasmid is very important in genetic engineering. So yes, they are also found in prokaryotic cells. Now the 70 Vedberg ribosomes, these are the ribosomes that are smaller in size. Yes, yes. In the eukaryotes, these 70 Vedberg ribosomes, where are they located, Farouk? In the mitochondria or chloroplast, and in case of plants. Prokaryotes, of course, that's the only organelle actually present in prokaryotes. So this is an easy question to start with. And then here, they are telling you, or they're showing you, two adjacent animal cells, cell 1 and cell 2. And they want you actually to draw, and the space provided, draw a diagram of the region R, and your diagram should show the 4 dark lines. So here, 4 dark lines. This is a phospholipid bilayer also, of course, and this is another one, and this is the space between them. What is the space between two cells? Intercellular space, tissue fluid. Okay, because here they ask you to draw space for diagram, and this is out of 3 marks. How will you draw this, the phospholipid bilayer? You don't have to draw with all the structures, but what will you draw?
Yeah, phosphate head.
The what? Yes, the phosphate head. Very good. Opposite each other, and then a space, and then, yes, very good. So this, how do you draw that? I'm going to draw phosphate heads, and then fatty acid tails, and then another. This is this. And then there's a small space, and then I'm going to draw it with me. You don't have a hard copy? You will do the same thing. And they said label. Didn't they say label? So you will label everything you can label. This is called the phosphate head, fatty acid tails, and this space is not the intercellular space, or the intermembrane space, because some students, I gave it as an exam to a bunch of students, and one gave me an answer, intermembrane space. No, intermembrane space is when we have a double membrane, and this is within the membrane, but this is between 2 animal cells. So like Hedy said, this is the tissue fluid. The tissue fluid or intracellular space, both will be accepted. Clear? So, okay?
So, now,
layer تاني فين؟ If it's double membrane ممكن تعمل layer تاني هنا، مش غلط. بس هما they just want this. Okay? The 4 this is one phospholipid membrane. This is one phospholipid bilayer. This is another phospholipid bilayer. Okay? And they have here the space between the 2 cells. If you're confused, correct, also accepted. Okay, because this is supposed to be, they did not say exactly 2 adjacent, so this is supposed to be the cell membrane, which is a double membrane. So the right thing is that you will have the double membrane here with the intermembrane space, but maybe it's not visible under a microscope. Maybe that's what's visible, the whole thing. Okay, because the mark scheme says that you only draw one phospholipid bilayer and another phospholipid bilayer and the tissue fluid. Okay, so let's look at the mark scheme so you feel that you're okay. Always use this side. This side is very good. We said this is March 2023. Yes, March 23, variant 22. So, dark line or lines labeled as phosphate heads, clear areas between the pairs of the dark lines labeled as fatty acid tails, reject if pointing to the intercellular space, clear area between the two cell surface membranes labeled as the interstitial fluid or the tissue fluid. So, and intercellular, so you will draw it. The dark lines, phosphate heads. These dark, the whole, not, what's your question? They told you draw what this line is. They drew it as a line. So you will draw it as a line again. They told you in the question, draw what is in that line. Okay, so you will have to draw the phosphate heads. Fine. And they will accept more or less any drawing you made, whether a phospholipid bilayer or the correct, which is a cell membrane that is a double membrane actually. So you have a phospholipid bilayer, intermembrane space, and a phospholipid bilayer. No, you have to draw the heads. Okay, where am I? Where is it? Where did it go? Yes, it's here. Fine. So you will have this space and you will label. Mitogens are short chains of amino acids that function as cell signaling molecules. Mitogens are released from secretory cells and travel in the blood to target cells, where the mitogens bind to cell surface receptors. The target cells respond by processing from the G1 phase to the synthesis phase of the mitotic cell cycle. So here, this is cell signaling that will stimulate mitosis. Do you understand the question? So outline what happens in the G1 phase and the synthesis phase of the mitotic cell cycle. So this is the lesson mitosis. What happens in the G1 or the growth phase? Yes, the cell will grow. Not only that, what else? Just discuss a little bit. What grows? What is being produced? Proteins are being produced. Give examples. Yes, give any example. We are making enzymes, we are producing RNA, we are growing, we're increasing in size, we're producing organelles. Okay, now the synthesis phase, what is mainly happening in the synthesis phase? Very good. Any replication of DNA, always give it its right name. The semi-conservative replication of the DNA. What else is being replicated? The centrioles. Okay. And between one phase and the other, there are always checkpoints. We will check that every phase is fine. Yes, check for any errors before we move on. You want to look at the mic scheme? As a result of mutation, the production and release of mitogens into the blood can be greatly increased. Suggest a possible consequence for target cells of increased concentrations of mitogens in the blood. Increased concentrations of mitogens. What will it do to the target cells? What did you say? I did not hear you. Yes. Okay. It will bind with complementary receptors and cause uncontrollable mitosis. Kareem, give me the phone. Give me the phone. Okay, in the growth phase, we will produce enzymes and blah, blah, blah. Yes, and synthesis phase, semi-conservative replication. Okay, you can also discuss when it is replicated, how does it look like? Uncontrolled increased mitosis. Okay, very, very easy up to now. We're doing a good job. Where are we? Cysteine is an amino acid that contains sulfur. Figure 2.1 shows the structure of the molecule formed by joining two cysteine molecules together. Draw a circle around an R group in the molecules. Now here you need to know the molecular structure of the amino acid. The amino acid is made up of a central carbon with two functional groups. This is one functional group, the amine group, and this was the carboxylic acid group, but it bonded with another amino acid. What is this? Where is the R group? You tell me. Yes. How many R groups do you have? I have this one R group, and this is another amino acid, the central carbon, the amine group here, the carboxylic acid group here, it's there, and this is another variable. So this is one R, and this is one variable group, and this is another variable group. They're called the side chains. Okay? Excuse you circle both. Okay? They said one? Okay. Draw a circle around an R group. Let's look at the mark scheme. Where's the mark scheme? Here. Both. Okay. إذا تمام برافو. Good. نروح فين؟ Fine. Now, where are we? Goblet cells in the human gas exchange system produce proteins called mucins. You take this in A2. The ends of mucin molecules contain many cysteine residues. Mucin strands are formed by joining the ends of the mucin molecules together through covalent bonds between the side chains. Name the name of these covalent bonds. Bravo. Because this is the only amino acid that contains sulfur. So this is the disulfide bond. You're with me? Okay. Mucin strands are transported out of the goblet cells and then absorb water to form mucus. Suggest and explain how the mucin strands are transported out of the goblet cells. Okay, 3 marks. Discuss exocytosis. Exocytosis. Excuse me? Very good. These vesicles, where do they come from? The Golgi body. So elaborate. The exocytosis means you have the cell membrane, the vesicle is going to come and fuse with the cell membrane and release its contents outside the cell. This is an energy consuming process. First, you will talk about the vesicle that will contain the mucin. It's coming from the Golgi body. So it's going to pinch off the Golgi body and then it will fuse with the cell membrane and it's going to release its contents. It's an energy consuming cell. And the movement of the vesicles, any movement inside the cell. Exactly. This is actually what I was really looking for, called cytoskeleton, which are made up of microtubules. So the movement, any movement inside the cell is helped or done by the cytoskeleton. So you will mention Golgi body, you will mention vesicles containing the mucin, they will move by cytoskeleton, they will fuse with the cell membrane, they will release their contents, you will mention the keyword exocytosis, and it is an active process. Write it down. لا مش لازم. More details are not wrong. If you're sure, write it. تمام؟ بس this مش لازم. Let me look at the mark scheme also. Fine. So disulfide correct. Exocytosis is underlined, so you have to mention it. And the steps of exocytosis. Vesicles that will contain mucin, they come from the Golgi body, they move by the cytoskeleton, they fuse with the cell membrane, it requires energy, okay? Mucins, they cannot cross the cell membrane because they're polar or they're hydrophilic. So we mentioned, how many marks? 3 marks. I think we mentioned more than 3 points. Okay, Yassine, put your phone away. Oh, you need a hard copy. Okay, because in any exam, you will always have a tricky question highlighted. Okay. Cystic fibrosis is a genetic disease caused by a mutation in the human cystic fibrosis transmembrane gene. And this results in a mucus that is thicker than normal. Suggest how thick mucus interferes with the maintenance of the healthy gas exchange surfaces in the lungs. Now, this is 2 marks, but you can write a lot of things. Very good. So this mucus is too thick and it will not be removed by the what? Very good. It will not be removed by the cilia. So it will accumulate. And what does it cause? It will cause inflammation and infection. And you can write the story that follows. It will attract what? Very good. It will attract phagocytes because phagocytes are always attracted to the site of infection. Phagocytes, in order to create a pathway, they release an enzyme. Remember, you were with us when we explained this lesson, the elastase enzyme. So the phagocytes, in order to reach the site of infection, they release the elastase enzyme to create a pathway. And the elastase enzyme, Farouk, from the name, what does it break down? The elastic fibers around the alveoli, preventing the alveoli from stretch and recoil. لا ما فيش mucus في الالveoli ما هي it will accumulate inside the lung. This accumulation of mucus causes infection.
يعني هو في ناس كويس بره الألفيول.
حبيبي الميوكس will accumulate here in the bronchioles and the bronchi. كمان it can accumulate inside the alveoli وتعمل blocking للairways. Okay? احنا قلنا حاجات كتير. We said a lot of things because it's only 2 marks, but that same question can be out of 4 marks. Asking you for the whole story, okay? It will not move, pathogens will build up, more chance of infection or disease. So we mentioned the story because that same question can be out of more marks. Okay, I don't like these questions, but we cannot help them. Row 1 and row 2, Kareem, of the table show the DNA-based sequences of part of the normal gene and some part of the mutated allele. The base sequences shown are for these strands used in the synthesis of RNA. When table 2.1 is completed, arrow 3 will show the base sequence of the RNA that came from the mutated allele. So of course, they will ask you to find the complementary. This is the mutated DNA and the complementary RNA. T is what? A. A, U, this is RNA. C, A, U, U, G, G, A, U, okay? G, U, U. Now, definitely there is a mutation, and they will ask you which type of mutation is happens here. Can you spot it out? Huh? Yeah. All this base triplet has been deleted. Did you notice that? And this has been shifted. All this or this, we have 1, 2, 3, ACC. This was shifted here. Did you see the ACCA here? ACCA, it has shifted. How did you know that this is a deletion mutation? Because there is a mutation from the point onwards. If it was a substitution mutation, only one base will be substituted or one base triplet will be substituted. But actually, from starting here, we have a messed up sequence of bases. Bardo, it's going to be a frame shift. Addition or deletion will cause a frame shift. Insertion. Okay, addition also is accepted. Fine. So the types of mutations, remember them: insertion, deletion, substitution. This substitution can either be silent, remember, or it will give you a premature stop nonsense, or a different amino acid altogether, the missense, remember? So here, what happened is that those 3 were deleted and those were shifted. So this is a frame shift mutation. It has a knock-on effect, remember? Okay. So the difference between the DNA-based sequence in row 1 and in row 2 is caused by a single gene mutation. What is the name of this type of mutation? Deletion. Abdullah. Okay, let's just make sure. Yes, it's deletion. Row 1 and 2 show the DNA strands used in the synthesis of RNA. State the term used to describe the DNA strand that is used in the synthesis. Template, very good. Complete table 2.1 to show the missing bases we just did. Didn't we?
Yes.
Okay, good. The normal allele is approximately 189 base pairs in length, and the mutant allele consists of only 1,480 amino acids. Explain the reasons for the difference between the number of base pairs and the number of amino acids. Okay, I want more answers. Do you understand the question? I have so many base triplets, and I have small number of amino acids. Exactly. Okay, I want 3 reasons. So I want you to mark this question. This is the question that I tell you is in every exam that is not nice. So the normal allele, Yasin, is what's this 1 million? Whatever base pairs in length. But the final protein is only 1,480 amino acids. So I have so many base pairs compared to the amino acid. Why? And I want 3 reasons. You already gave me 2 reasons. Hedy gave them to me. He told me that, yes, I will remove them. Okay, very good. So you gave me 3 actually, thinking together. Hedy said that the base triplet is degenerate. Who will remind me what is degenerate, Adham? No. Universal is something and the degenerate is something else. Yeah. Exactly. The same base triplet can code for the same, different base triplets can code for the same amino acid. So I can have several base triplets, but the same amino acid. Fine. What else? We have actually 4 bases with 3 possibilities, 3 different base triplets. We have a total of how many base triplets? 64. We have 64 possible base triplets, even though we only have 20 amino acids. I'm not here to joke. Okay? Even though I have only 20 amino acids. So different base triplets can code for the same amino acid. This is one reason. I want another reason. Hedy, you said it. Huh? Yes, some base triplets are non-coding. Some base triplets are non-coding. They have a name. Excellent. We took it in post-transcriptional modification. Remember? The messenger will take a copy of the DNA. It will copy the whole DNA. Some base triplets are non-coding. They don't code for proteins. So they have a name. They're called the introns. We did not study mitosis, even though we had a quiz. This was in protein synthesis. They're called the introns. And we said, how do we remember them? Because the exons will go out and the introns will be removed. Remember? And this is called RNA splicing. Post-transcriptional modification. I'll take a copy. I will make a messenger RNA, but I will modify it a little bit before I go out of the nucleus. So this is another reason. Some base triplets are non-coding. Give me, Farouk, a third reason. Maryam said it. In the final polypeptide chain, there was a base triplet that coded for the first amino acid, and it's called the methionine or the methanone. We remove it when we modify. Remember the post-translational modification? You were not with me when we took the cell structure. We will make the sequence of amino acids. And then this will go to the Golgi body or the rough endoplasmic reticulum to modify. I'll fold it. I will change it to a secondary structure, change it to a tertiary structure. I'll add a carbohydrate. I will add a fat. I will remove the start amino acid. It's not needed. It's called the methionine. I will remove the final amino acid that was coded for by the stop codon. I don't need it. Okay, I will create bonds and fold. These are the 3 reasons why I have many bases, but a smaller number of amino acids. Yes, you can also say that. This is what you will start with actually. That the base triplet 3 bases, 1 amino acid. So this is one reason. And they can have, you have different bases for the same amino acid. And sometimes they're not coding. So I have them there, but they don't code. Okay, they don't give me amino acids. I remove the bases because they would give me amino acids that are start or stop. Okay, mark this question, highlight it. Let's look at the marks. I think I answered it before. I answered it before with a bunch of kids. I saw this, yeah. And I looked at it yesterday. Where's the mark scheme? Idea of 3 bases coding for 1 amino acid. What just Hedy said it. Introns are removed in gene splicing. Introns are non-coding. Only exons are joined together. The DNA triplet or the messenger RNA codon for stop is removed. Methanone at the start is removed. Any valid point. Enhancer sequences and the terminator sequences. I think you take the promoter in A2. I don't expect you to know it in the AS. What's the promoter? What attaches to the promoter, huh? Yes. The messenger RNA binds to it. Okay. But في any valid point? Okay. Good. Can you be quiet or it's impossible? Now we finished this question. No, this is my solving. Okay. Explain why cell X and cell Y have very different appearances in this transfer section. Both of them are which type of cell, xylem or phloem? Of course, how did I know? I have a companion cell next to it. Why does this look like this and why does this look like this?
Why is this the same?
What's the relation between the cell? I'm looking at it under the microscope. I see sieve plates and in the other one I see it empty. Why? Yes, because I took it from different levels. Like, for example, the phloem cells, they are like this. I have a phloem cell, okay, with a sieve plate. And the one next to it, for example, is here at different levels. So when I cut this specimen and I look at it under the microscope, here it was at the level of a sieve plate, here it wasn't. Okay? If this question, I think I told this question before. So here we're just at different levels. That's why you see it in one of the specimens in Y and you don't see it in X. Let's look here. Cell Y shows a sieve plate. Idea that the sieve plates are different heights in the stem. Okay? So one of the cells shows the sieve plate. Abdullah, another cell does not show the sieve plate. Easy? Okay? These are easy questions. Up to now, only one question that I thought was not. نعمل ايه طيب؟ هم عايزين درجة يعني because you will mention one sentence on X and one sentence on Y. Okay? Because you saw it here and he mentioned the word sieve plates and phloem and whatever. Okay. Sucrose is formed from the glucose synthesized by mesophyll cells. Explain how companion cells are involved in the transfer of sucrose. I explained it today in detail. Write it. Write it. Okay? We said we will talk about the hydrogen pump and we will talk about the co-transporter protein and the transfer of sucrose into the phloem sieve tube element. And then we will say the apoplastic. And why are you looking at me? Can you solve it? تفضل. Yeah. You will talk about the hydrogen pump and then you will talk about the co-transporter. Oh, هي الاثنين تحت. The same name. Okay? Now, where is it written? If you want word by word, Yani. I get you. Let me open this the solved version. Okay. This is here snipped from the notes you study from. Okay. And the mark scheme, I was very happy to see that all marking points from an annotated diagram are accepted. So in any time you see that a small diagram will help you in your answer, go ahead. They accept annotated diagrams. Pencil, pen, whatever, but pencil is fine. Excuse me? Oh, it's some lines. Oh, all marking points from an annotated diagram. You will talk about the pump and the co-transporter protein and all the steps and everything we said today while we will explain it in class. And you can draw it. And I told you, always draw while you're studying about it. Studying about, you always have a paper and a pen and draw things because they help you remember. How will you draw that? You will draw the cell membrane, label that this is cell membrane, and this is the cell wall, and sometimes the cell wall is just not with a line. This is the cell wall. This is the cytoplasm. Okay, if you want to draw an annotated diagram, this is the phloem element with plasmodiesman. That's it. Of course, yes.
It's just great for the nitrogen, so transportation by secondary active.
Yes, the sucrose by secondary active transport hydrogen by facilitated diffusion. How do I draw the two proteins here? Right answer. I will draw this as the hydrogen pump. And the co-transporter protein, I will draw a protein with two depressions, like this. Okay? Here I have the cell wall. I have a buildup of protons, proton gradient. and then it will move down the co-transported protein by passing liquid diffusion. And with it, I will have these sucrose. An annotated diagram like this is very, very good. Okay? And then the sucrose will move through the plasmodesmata by diffusion. So this is an annotated diagram which will give you grades because you are explaining through the diagram. You can actually start with it. If these are your lines, you can start with the diagrams here, taking a small space and then writing the answer below it. Perfect model answer. Okay, Ada? Can you write the answer? You're too lazy? Write it down. You did? Repeated where? Where did we repeat it? Okay, draw the diagram. At least draw it. Noor and Kareem, stop talking. No, don't discuss anything. Okay? Explain how hydrogen bonding occurs between two water molecules. We explained it to you. Yes, yes, yes. Okay, I want you to explain this using a small diagram also. How will you draw it? We just did it today when I was explaining. Yes. If you don't want to write, you're too lazy to write, and please do the diagram. Okay? Because a dipolar molecule, the O bonding to two hydrogen, and don't forget the symbol. The electro negative, electro positive, electro positive. It's called dipolar. And then another water molecule, electronegative, electropositive, and this is the hydrogen bond here. It is represented by dashes. So if you don't want to write it down, put the dashes. This is the hydrogen bond. And two similar molecules, this is cohesion. This is amazing. Believe me, the examiner will be very proud of you. Okay? And discuss another two sentences, that. Yes, go where? No. Eh? Should it? Where record? Now it is recorded that you want to go to the bathroom. Okay, Kareem. Good morning. How hydrogen bonding is involved in water transport and the xylem of the plant stem. Yes, write it down. Let's at least write it points, cohesion and adhesion. It's as if they are repeating what we explained today. Do you want the answer? No, it's not the answering to it. It's correct, but they did not ask about it. So you have here the answer in detail. Okay, if you want to write exactly what, and the transpiration pull and all the keywords and the capillary action and to prevent the water column from breaking, etc., etc. Okay, Ada? Okay, you will not write it down? You need the bathroom desperately? Now everyone knows. Adham. No one knows you. Like they won't know which Adam, you know. What's your second name? You're writing or you're wasting? Or... Kareem, where are your past papers? So you have them and you did not bring them. No, this is too much. Seriously, this is too much. Like you have them and you forgot them. Like you're coming today for a biology class. I don't see a copybook. I don't see notes. I don't see classified. I don't see past papers. And you know that we're solving past papers. Seriously. I want to finish this exam. We're not leaving except when we finish at least one full exam. Hydrogen bonding again between water molecules give water a relatively high latent heat of vaporization. Wow. We just explained it. Yes.
To allow water molecules to change from liquid.
Very good. I need to absorb lots of energy. And this is going to give me cooling effect. This is what I was looking for, okay? You'll study this in the physical properties of water in the lesson biological molecules. Fine. Now, everything here, what Noor said it in detail. Plant leaves when they heat up in photosynthesis, they need the cooling effect to prevent the denaturing of the enzymes. Also, we said it today while we will explain. Can you write it? Write it at least in points, quickly. You wrote it also? Okay. With all the details and the keywords? Doesn't look enough to me. Okay, Farouk, go on. Heidi, where are your past papers? You have them? No comment. And I will solve June. We're leaving at 10. Because June is easy and it's important. The lesson? They're not coming. Okay. Wait, what's tomorrow? I can have a class on Thursday. We can solve with this exam, the other exam. No, we'll finish this today. We'll finish it today. Okay? We are already in page 11 anyway. Explain what is meant by infectious disease. 2 marks. The same disease, the same definition in O level. Yes, and it can be transferred. And give any example. Any example. Tuberculosis, cholera, any example. Name a species of organism that causes TB. Or tuberculosis. They always ask you because you have 2 here causative organism, Mycobacterium bovis or Mycobacterium tuberculosis. Which one causes it in cattle? The bovis. Name the type of immunity that a mother gives to her baby through her breast milk. Passive and artificial or natural? Natural. So natural, passive. Okay, explain why different antibodies need to be produced to give immunity to these new strains. You have new pathogens.
Pathogens have different.
Okay, okay, yes, you're right. Part of the answer. We have new pathogens and we have different antigens. Very good. Bravo. Perfect. We have different antigens of different shapes and they bind to different antibodies that are complementary. Yes, whatever. Okay, that are complementary. Wait, how many marks? What if this question was out of more marks? No. We want to remember together the structure.
في ناس عندها افضل شو بس عشان نعرف نصير نسمع ممكن اقول اسمي
اتفضل.
عمر محمد فاروق محمد فاروق بس عمر محمد موجود الان
Then he's not in class. يعني he's here بس ما جاش. فاروق عنده after school. ده فاروق جميل. كريم ما عندوش after school. متأكد؟ متأكد؟ Okay, if this question was out of more marks, Kareem, can you remind me of the structure of the antibody? Yes, but what is the main structure of the antibody? Yes, 2 long chains. Draw this with me. And this can be an annotated diagram. What? 2 long chains or heavy chains and bonded to 2 light chains. And what's the bond here? Disulfide bond. This part is sometimes found, sometimes not. It's the hinge that gives flexibility. Okay, if it is bonded to the phagocyte here, so here is the there is a complementary receptor that will bind. This is the part that bonds with the complementary receptor on the phagocyte. And what's this part? The variable region. They like the variable region. We have 4 variable regions and 2 antibody antigen binding sites. Kareem, this is the antigen antibody binding site. And each one of them is a variable region. This variable region is variable because it has a different sequence of what? Amino acids, and it's unique for every antigen. That's why we have different antibodies for different antigens, because we have different variables. Okay? Four variables is 1, 2, 3, 4, and this is an antigen antibody binding site, and 2 antigen antibody binding site. Okay? Good. So I have children here. So where are we? This is the answer from the mark scheme. Because like Hedy said, there is antigenic variation. The antigens will variate and they will mutate. So we have to create different antibodies every time the antigen changes. And the word complementary, we said it, and they're specific to different antigens and the structure. These are the variable regions, Hedy. See, this is one. So you have 4. And this is the antibody antigen binding site, and this is the variable region 1, 2, 3, 4. This is the hinge. These are the disulfide bonds, and this is the part where they bind with the complementary receptor. Okay? We'll finish this exam and then I'll let you go. Explain how vaccination programs can help to control the spread of infectious diseases. Okay. What? Yes. Now, vaccination is very important, whether an O-level or AS. Why does it fail? Why does it succeed? How does it protect you? Okay, that part of the making of the memory cells and the success of the vaccination programs. How does it help to control? Now, we will just say herd vaccination, that's it. What else do we say? We will mention herd vaccination. Kareem, we will break the transmission cycle. We will prevent the pathogens from spreading, because we will have lifelong immunity and we will create memory cells that will divide quickly by mitosis upon a reinfection with the same pathogen. They will prevent the transmission of disease. We might need a booster dose to make or to strengthen the immune system. Let's see, what do we write here? A lot of things. Vaccination program, 1 minute. Long-term immunity, vaccination individuals, the answer is there. Always, why a vaccination program fails or succeeds, it's a very common question. Okay, let me get you the answer from the mark scheme here, because it's very common. Vaccines are very important. Whole population herd to have an immune response. We said the long-term immunity and the memory. Artificial active, we did not say it, we said memory. Cannot transmit the virus, will prevent herd vaccine, we said it. Any valid point refers to vaccination programs aimed to break the transmission cycle.
Okay.
You tell me. We took it in the lesson. Why vaccination programs, they sometimes fail? Huh? Yes, sometimes we cannot make vaccines to a virus like to a pathogen like HIV because it changes. And if you take the vaccine, also it will fail. Why? Huh? Yes, the opposite of this. Sometimes we need a booster dose and people doesn't take it. Sometimes we don't achieve herd immunity. Sometimes it's expensive. Sometimes people will not give a positive response. It will give you an allergic reaction. It needs to be frozen or transported in a special way that is expensive. Sometimes we have wars or natural disasters that will not help us succeed in the vaccination programs. It's a list. You have a list, successful vaccine. Why? Why does it succeed? Why does it fail? It's very common. Remember, study the list, okay? Let's finish. Explain why penicillin is not effective against viruses. Oh, I gave you the answer. Yes, the virus, because the antibiotic either destroys the cell wall or prevents protein synthesis or prevents reproduction. The virus doesn't do all these things. Yes, it's not dead. It's not considered living, except when it is inside a host cell, and it will only reproduce then. Okay, it doesn't have a cell wall. It doesn't have metabolic reactions. It is non-living. It does not reproduce except inside a host cell. This is an O-level question. I will not. Okay. Complete the figure to show how the machine is connected to the right atrium and to the vena cava using a single line to represent each cannula. You know what's a cannula? You have to read the question, and they tell you that pneumonia is a severe lung disease that will interfere with the gas exchange. A person with pneumonia can be connected to a machine. This machine will perform the gas exchange, and the tube is inserted to the right atrium, and it will take the blood from the machine to the pump. And then, listen, this is a question that you have to read carefully so you know how to answer. And then, when the exchange occurs, the blood is then warmed and returns by another cannula to the vena cava. So, this heart is connected to this machine. It is connected to the right atrium, and then from the machine to the vena cava. So they're asking you, or they want to know if you know the right atrium and the vena cava and their positions. Yes, so it's the right atrium here. This is the right atrium. It will move from the right atrium to the artificial pump, okay? And then from the pump to the vena cava. Where's the vena cava? Yes. Just one arrow. Okay, Noor? Clear? State the name of the structure and the gas exchange system that has the same function as the partially permeable membrane of the oxygenator, which exchange gases. Where is gas exchange taking place? Alveoli. Can you be silent and let's finish? Yes, we will finish. Suggest and explain how the oxygenator carries out the functions of the gas exchange normally occurring in the lungs. So you will talk about the adaptations of what? What did we say in the first answer? Alveoli. Alveoli does gas exchange, and we have adaptations of alveoli. Yes? You need to mention two factors: the direction of the movement of the gases and the adaptation. Oxygen will move from the alveoli to the surrounding what? Capillaries by diffusion. And the CO2 will move in the opposite direction. Why? Because of the concentration gradient. You will mention this part. And then the adaptations of the alveoli. Mention the adaptation. One cell thick. Huh? Large surface area.
Has gaps.
It has gaps.
Moist.
It's moist because we have surfactant to prevent its collapse. Okay, to reduce the surface tension. It has elastic fibers to stretch and recoil so that I can expel the air when I exhale. I have many capillaries to maintain a concentration gradient. Okay. You're not writing it down. Then what? So what do we say here? We will mention the directions, any three, and then the adaptations. Oxygen will move from the air into the surrounding capillaries, and carbon dioxide from the blood into the whatever by diffusion down a gradient. Maintains a steep diffusion gradient, referred to equilibrium is not reached. Short diffusion pathway across the membrane, any valid point. We said a lot of things. We also said large surface area. Okay? I don't know why you look bored. Well, the oxygenator is taking the place of the alveoli. Okay? So see, here the oxygen, the whatever, the oxygenator is in replacement of the alveoli. Fine? Okay, explain how the structure of the tunica media in this figure is different from the structure of the tunica media in a muscular artery, and relate the difference to the function of the aorta. Very good, because that's an elastic artery to withstand to withstand the blood pressure, to smooth out a pulsatile flow. So the aorta is a elastic artery, more elastic fibers for the reasons Hedy said, to withstand high blood pressure, to smooth out pulsatile flow, so it doesn't rupture, so blah, blah, blah. Here, it contains more smooth muscles. Why? And control the diameter. Very good. Explain why the physiology of this requires carbonic anhydrase. What is the carbonic anhydrase? What do we do with it? Yes, to catalyze the reaction, and you write it down balanced. Okay, Farouk? Explain why whatever does not require the chloride shift. Now you have to be the question. The chloride, why does it go out? Why does it come in? To prevent a buildup of charges and to make the cell neutral, yes? Here, there is something that will not do the chloride shift. There is a reason why. The hydrogen carbonate ions will combine with hemoglobin here. So it will not go out and it will not disturb the charges. You finished? No. There is a question.
They move in many directions. So
Yes, collagen, very important. We have so many reasons why it is strong.
they move in many directions.
Yes, we have the triple helix, strong hydrogen bonds. They are staggered, covalent crosslinks, moving in different directions. They have fibers and staggered ends.
Each third amino acid with vaccine, so they can coil.
Yes, they can come very close together and make a tight coil. So tight coiling, many hydrogen bonds, staggered ends, covalent crosslinks. They move in so many different directions to make the fibers so that they withstand stresses. So we said here 5 reasons. The enzyme collagenase breaks down collagen. Collagenase is this is a graph. What do they want here? Explain why the activity of the collagenase is lower at this pH than the optimum. Now, this is an O-level question. Hydrogen ion? You mean it will be more acidic? What will this do to the enzyme? Yes, and you will discuss. Wait, no one is standing at the door, and there is no lesson. Yes, mention that the active site will become destroyed and will not be no longer complementary. You cannot do an enzyme substrate complex. I didn't finish. Yeah. Wait, the exam is finished? There's a blank page. Okay, when will we have an online? Let's explain and revise.